All right, back for another video. This time it will be on an introduction to the autonomic nervous system and a little bit of its anatomy. So we'll talk about sort of the wiring of the system, focusing on the motor components. We'll talk a little bit about the sensory input that leads to the motor output, but we'll talk mostly about the motor output, the neurotransmitters involved, and the target tissues. So if you recall from a big intro to the nervous system, where we divvied up the nervous system into central nervous system and peripheral nervous system components, the ANS, the autonomic nervous system, we also called it the visceral motor system. Remember, we had somatic motor, which is control of skeletal muscle. Visceral motor is control of a different set of target tissues. In this case, the target tissues are, and these are important, cardiac muscle, smooth muscle, found in a whole variety of locations. Remember, smooth muscle is found in the walls of hollow organs, found in the walls of blood vessels, in the walls of bronchioles of the lung, in the walls of the GI tract, in the walls of the reproductive tract, and on, right? Again, the hollow structures there contain smooth muscle. And the last target tissue, well, actually there's two, I guess, are glands, largely exocrine, but also some endocrine glands are controlled by autonomic, have input from autonomic nervous system. And then adipose tissue has a little bit of autonomic control, right? So cardiac muscle, smooth muscle, glands, and adipose tissue. All right, so a little bit about the function, right? We said it's visceral motor, but the idea here is that, right, we need to control systems that help us regulate homeostasis. And if you recall, homeostasis is that regulation of the internal environment. And again, I made that a little bit more specific to sort of clarify it, to the interstitial space with which all cells need to exchange to remain alive, right? They need to exchange oxygen and nutrients and waste with the interstitial fluid right outside their membranes to stay alive. Well, our organ systems, in many cases, are adapted to maintain that environment for the cellular exchange. Well, if you think about it, blood flow is a good example that needs to be maintained because the interstitial fluid exchanges with blood, right? And the cells exchange with the interstitial fluid. So maintaining blood flow to all tissues, or almost all tissues, there are a few avascular tissues, but maintaining blood flow to tissues is essential to homeostasis. And the understanding, and again, this is a lot of AMP2 stuff, but understanding blood flow regulation is a lot about understanding autonomic control of cardiac muscle and smooth muscle found in the walls of the blood vessels. And we could go on and do examples from respiratory system and other systems as well. Okay, so let's look at the autonomic nervous system in terms of, we know its major targets now. It's actually divvied up into two main components or divisions. And those divisions are the sympathetic division and the parasympathetic division. I'll start to write some of these things down. Right. So ANS is divvied up into sympathetic nervous system and parasympathetic nervous system. And you could just say divisions if you prefer. Now the sympathetic division, the sort of the phrase that goes along with that is the fight or flight system. And the easy way to remember that is that it is the system that allows you and promotes homeostasis during fight or flight. In other words, during physical activity, right? The fight or flight is sort of an evolutionary term like what animals need to do, in a sort of stressful situation, right? But for humans, and we can have that same situation in humans, but a less extreme situation is just anytime somatic activity goes up, i.e. exercise, increases, we're going to get an increase in sympathetic output because it is going to function at those target tissues to help maintain homeostasis. The parasympathetic, excuse me, the parasympathetic division, and its phrase that goes along with it, descriptive phrase, is rest and repose or rest and digest. And so it's a system that is adapted more so to digestion and then the promotion of nutrient storage and anabolism. Anabolism being the building of molecules and structures. And again, so if we promote digestion and we're promoting growth and repair through the parasympathetic division. Now, at many targets, the sympathetic and parasympathetic, so the innervation from these two divisions, overlaps. Right? Not all, but most target tissues have both sympathetic and parasympathetic. They tend to be antagonistic. They tend to sort of be the gas and the brake at those two. And again, one could be which one's the gas and which one's the brake, depends upon the target tissue. But they tend to be antagonistic at those target tissues. There are a few systems that are innervated by only one of the two divisions, right? And by the way, these are what are called closed loop motor systems. What that means is that we're going to get, and we talked about this is a homeostatic loop. That's a closed system motor control system or motor control function. And what it means is that what the homeostatic loop, what the sympathetic control system is controlling, that parameter feeds back as input to the control system to adjust the output of the control system. So in other words, if blood pressure were to fall, that would be a stimulus that we might want to increase cardiac output. Increasing cardiac output would increase blood pressure, solving the problem. That would feed back, that measurement of blood pressure would feed back and turn that system off, right? So closed loop systems are modified during the action and continuously during the action. So as blood pressure goes up and down, we continuously receive information about our blood pressure and whether or not the system mechanisms that we're using to control it are working. And if they aren't, then the stimulus remains and and may even get bigger, such that the output becomes stronger and we try harder and harder effectively to solve the problem. Okay, let's look at I think we've got the basics, right? The two opposing, again, usually opposing systems at these target tissues and their specifics. We'll get more into much more detail about what the sympathetic is promoting specifically versus what the parasympathetic is promoting or inhibiting specifically. But right now in this video, I just wanted to give the introduction and a big chunk of the anatomical description of each of these systems so we can understand how they're interacting with their target tissues. Okay, so to do that, we need to draw the central nervous system and then the essential components of the peripheral nervous system that we need to lay this out. So I'm going to draw brain and spinal cord, right? So we'll just put a B for brain there. And then SC for spinal cord, right? So that's our central nervous system, right? Lots going on in there. There is a main center for autonomic control. It's in the hypothalamus. And there are multiple inputs to that center that help us control the output through this autonomic division. Some of them can be initiated within the brain itself. The limbic system, for instance, is tied to emotion. Obviously getting scared, right? Think about watching a scary movie. Can make your palms sweaty, can make your heart rate go up, right? That's all autonomic output would be centrally generated stimulus. We can also have peripherally generated stimuli, which of course would be through sensory neurons. And again, now remember when we talked about sensory division, the afferent division of the peripheral nervous system, we divvied that up into special senses, right? Vision, hearing, taste, smell, equilibrium, right? We divvied it up into somatic senses, right? Proprioception, where your joints are, like tension on your muscles, things like that. Pain, tactile, right? But then we also had visceral senses, right? And visceral senses, pain is also a visceral sense, but some of those visceral senses are adapted to sense things like distension of these hollow organs, right? Distension of the blood vessels, distension of the heart, distension of the bladder, right? Among other parameters, chemical composition of the blood would be sensed and used as information along these sensory pathways. And so we can draw sort of our general sensory neuron, right, leading into and synapsing with, remember that's the cell body, right, leading into the central nervous system. And I could have it in the cranial region as well. Now, the anatomy of the two divisions of the ANS, right? So again, I'm just being generic here and not putting in all the details, including the interneurons between them. But the sensory information would come in. Interneurons likely, information sent to the brain as well, although there are spinal cord dependent reflexes. In other words, they can come in and out of the spinal cord alone without going to the brain. But just to simplify, sensory neuron to interneurons to processing and then generation of a motor output along one of these two or both of these pathways. And so let's see, let's start with the sympathetic division. And the sympathetic division is characterized as thoracolumbar because the motor neurons that carry that sympathetic output out of the CNS originate in the thoracic, a little reason for cervical here, thoracic and lumbar, and lumbar regions. This would be sacral down here, right? And so, and if you recall from, again, day one, we talked about this, the anatomy of this very generally, but the autonomic nervous system motor output is a 2-neuron system. Remember, somatic motor was a single neuron, again, in the peripheral nervous system, single neuron, with the cell body found in the CNS, and then the axon extending all the way out to the skeletal muscle, right? Somatic skeletal muscle. For the autonomic, the sympathetic neurons are derived from the thoracic and lumbar regions, so I'll go down to about here. That might be a little too low. Whatever, but so thoracic and lumbar, right, we'll call this cervical up here. And those neurons would extend out into the periphery, and they would form a synapse with a postganglionic neuron. Now, many of those synapses in the peripheral nervous system are very close to the spinal cord. Right, so I'm going to do that. I have a bunch of them be very close here. There is one oddball that goes way out here. And there are a couple others that extend a little bit farther. They form what are called collateral ganglion. Again, that's more anatomically important than it is in terms of the functional anatomy I'm trying to convey here. So I won't draw collateral ganglion. They're just not that important. But they would extend a little bit farther out here before they form that ganglion. But I'm not going to get carried away with that. I'm going to change my color because the preganglionic neurons will synapse on the postganglionic neurons. Right, so this blue cell, right, cell body initially. Now I'm I'm going to make this one out here blue as well, but it's going to be a little different because it's not a neuron per se. All right, we'll get back to that one. Okay, so now I have the postganglionic cell that would extend all the way out to the target. Remember, these targets are all over the place, right? This is smooth cardiac muscle. That's obviously all in one location. But then smooth muscle found in pretty much everywhere. We have blood vessels everywhere, right? But also GI tract, reproductive tract, respiratory tract, right? Now, are any of those, do you have blood vessels in your head up here? Sure you do. Now, so what that means, and again, without getting carried away with the anatomy, is that we're going to have some branching of these preganglionics that run up, right? Run north and south and travel through the spinal nerves that exit the spinal cord and even the brain at all different levels, right? Because we have to innervate brain structures, we have to innervate legs and things like that, blood vessels in the legs, reproductive structures. And so it looks like they're all coming out of this thoracolumbar region and staying in that region. They don't, right? We get branching. So there is, divergence of preganglionic neurons, right? They split and diverge and synapse on a variety, up to 20 or more postganglionic. Now I'm showing it as one-to-one. I'm showing it looking like serial processing, but it's not. We try to draw all the complexities here. The picture will get really ugly really fast. But just to point that out is that it looks like the sympathetic output comes from just this region and goes to just the regions that these spinal nerves would innervate, but that's not true, right? We have to have this sort of branching and going to all possible targets, those visceral targets. All right, and then we can have our target cells out here. Right, which are, again, cardiac muscle, smooth muscle, glands, adipose tissue. All right, so in the sympathetic division and in the parasympathetic division, and I'll draw that in a moment, the neurotransmitter released by the preganglionic, so remember this is a ganglionic, so this would be called a sympathetic ganglion. I'm going to circle this one. It just says sympathetic ganglion. Sorry, I'm writing that a little too small. Have to keep that in mind. Right, that's a sympathetic ganglion, right? So that's where the cell body found in the peripheral nervous system, a clump of cell bodies, not one. Right, there'd be a whole clump of them here. And at that ganglion, the neurotransmitter released by the preganglionic cell is acetylcholine. Right, and again, I won't try to squeeze little dots in there, but I will label that as ACH being released. Now that acetylcholine obviously, right, acts as a neurotransmitter. It is a neurotransmitter. Diffuses across the synaptic cleft, binds to the postganglionic cell at receptors that are called cholinergic receptors. The cholinergic receptors bind acetylcholine, right? Now there's a variety of those, two main classes, right? And so acetylcholine binds nicotinic, that's the subgroup, cholinergic receptors. So I'll put that label here on the postganglionic cell. Guess what? We want, if we're activating this pathway, we want stimulation of EPSPs there and action potentials in the postganglionic neuron. So acetylcholine binds to nicotinic cholinergic receptors, right, which are on ionotropic channels, right? So acetylcholine opens ionotropic channels. Remember, ionotropic channels allow ion fluxes, and those ion fluxes, in this situation depolarize this cell so the ion fluxes would be sodium or calcium, right? And again, just going back to what we've already talked about in terms of this, the idea of gradient potentials leading to, or inhibiting in some cases, action potentials. Well, if we're activating this pathway, we want action potentials, which means we want EPSPs, which means it has to be sodium or calcium influx, because those are the only two ions that we've talked about that generate depolarization and EPSPs. Okay, so now, right, so if acetylcholine is released, it opens the channels, right, that are ionotropic and bind this, or are nicotinic in type. We're going to get an action potential along this end, or along this axon, the postganglionic axon, and we're going to release a neurotransmitter out here. Well, the neurotransmitter out here is not acetylcholine. It is norepinephrine. NE for short. Okay? Now, norepinephrine binds not cholinergic receptors. It binds what are called adrenergic receptors. Right out of room over here. I'll write it over here. So ACH binds cholinergic receptors, and they can be either nicotinic, or the one I didn't talk about yet is muscarinic. Okay, and norepinephrine, get down a little lower here, binds adrenergic receptors, and they fall into alpha and beta types, right? And they're actually subtypes of those types. Okay, so, or norepinephrine is released, our target tissues, cardiac muscle, smooth muscle, glands, adipose tissue, have adrenergic receptors of either the alpha or beta types, and sometimes both. Right? And so the actions at the target are mediated by norepinephrine binding to these two, one or the other or both, receptor subtypes at the target here. Okay? All right. Now, let's see. What we've left out is this strange thing right here, right? And what that is so this second, the postganglionic cell here is not a neuron. It's sort of it's the derivative of a neuron. It's called a chromaffin cell. These are cells found in the adrenal medulla. So this is the adrenal medulla being the inside layer, if you will, of the adrenal gland. All right, so we put sort of a inside layer here, and I'll make an outside layer. The adrenal gland's kind of pyramid shaped. There we go. Right? So that is the adrenal gland. Well, we did say that these systems can innervate glands. So in this scenario, though, this is like a direct. It's a component of the gland, essentially, right? Where this blue guy, the chromaffin cell, and that's not that important for now, actually releases epinephrine and norepinephrine. So there's the norepinephrine, but it also releases epinephrine, also known as adrenaline, right? So NE and epi for epinephrine. Well, the difference here, like this norepinephrine and that norepinephrine, it's the same molecule, But this is released at a synapse. This is released, both of these are released into the bloodstream. And what that means is that the sympathetic division of the nervous system, sympathetic nervous system, has an endocrine component. In other words, it's neuroendocrine in function. It's the linkage between the nervous system here and the endocrine system here that's going to release these two guys, these two chemicals as hormones. Are going to be carried in the bloodstream. They're going to be dispersed to almost all target tissues. Again, the blood-brain barrier tends to limit blood exchange of certain things with the brain tissue. But essentially, all other tissues are going to be exposed to these, which means that we get this very broad band, if you will, exposure to sympathetic hormones, right? The parasympathetic division is not going to have this kind of component. It's not going to have an endocrine component. So while these are very focal, and again, focal, yes, we had a lot of, divergence here. So we can definitely have a lot of target tissues activated with the sympathetic division. But this is going to make it even more so, because now these chemical agents, in this case hormones, have access to target tissues anywhere. And so any tissue that has an alpha or a beta receptor, even if it's not innervated, can respond to the sympathetic output. Okay, so let's summarize. Our sympathetic division is fight or flight. It's got short preganglionic neurons, most of them, and therefore long postganglionic neurons. It uses acetylcholine and nicotinic receptors at the ganglia. It uses norepinephrine at the target tissues, which binds on alpha and beta receptors. And it also has this hormonal component by innervating the adrenal medulla. It is also thoracolumbar in origin. So how does the parasympathetic division differ? Well, it is craniosacral in origin. In other words, it's going to come from the brain and then the sacral regions of the spinal cord. Now, the thing to remember here is that the targets are pretty much exactly the same, right? Remember I said we have dual innervation through sympathetic and parasympathetic divisions to most, not all, target tissues. And so the preganglionic neurons are going to come out of the brain or sacral spinal cord. And actually, most of the parasympathetic comes from one of the cranial nerves. It's called the vagus nerve. Vega refers to wandering. And so that nerve is going to actually wander around the body, essentially, right, to many different target tissues. And again, many of the exact same targets that the sympathetic division is innervating. So I'll have some of these. In the parasympathetic division, the preganglionic neuron is quite long, and the postganglionic is short, the exact opposite of this. So we'll have to have this guy come out and be reasonably long. Now, I'm going to make this one the vagus. And there's several cranial nerves that carry parasympathetic information. One of them, the vagus nerve, carries most of it. And actually, so that's the guy that tends to carry most of its information. I don't want to try to overlap it, but into the abdominal thoracic cavity where the organs are, right? Because again, a lot of our cardiac, all the cardiac muscle and a lot of the smooth muscle and glands and other things are found in the abdominal pelvic region. Not entirely, but a lot of our organs are in this area anatomically. And so this guy has to come down right into this region to innervate these targets. So lots and lots of branching, obviously. And now I'm going to do the postganglionic neuron as much shorter. And remember, there is no hormonal component. Oops, need some axons here. The you might wonder what the sacral region is innervating of the parasympathetic division here. Well, largely pelvic structures. So things like the bladder and the reproductive structures are innervated by the parasympathetic sacral component. Again, put my short postganglionic neurons. And our target cells, which again are the exact same target cells we have here. Again, mostly the same target cells that we have in the sympathetic division. Just be tough to draw them all overlapping and actually see what's going on. Okay, so now our ganglion is way out here, right? Relatively far out compared to the sympathetic ganglia. Again, with the exception of the collateral ganglia, which I said I didn't draw, but they're a little farther out, and then this oddball, right? But so long preganglionic, short postganglionic target cell. The nice thing about the parasympathetic ganglia is they look just like the sympathetic ganglia. So in terms of what's released and what they bind to. So it's acetylcholine and acetylcholine binding nicotinic receptors. Exactly the same thing that we saw here, right? Now, they differ at the target tissue. In other words, what does the postganglionic cell release onto the target cell? That differs. So in this scenario, it doesn't matter which of these I label, but in this scenario, it's going to release acetylcholine again, right? So it was acetylcholine here and here. Now here it was nicotinic, here it is the other subtype, which is muscarinic on the target cell. Muscarinic receptors. And again, there are multiple subtypes of the muscarinic receptor. It's probably important to point out that the multiple subtypes, right? Because really, if you think about it, we've got acetylcholine, epinephrine, and norepinephrine as our neurotransmitters in these two divisions. And we have a whole huge variety of targets, right? And again, it sounds like a small number, but think about smooth muscle in the blood vessels versus the bronchioles versus the reproductive tract versus the wall of the bladder versus that. We can go on and on, right? And so how do you get this very small group of neurotransmitters to generate effects that maybe differ at different locations? And the answer is differences in receptors, right? In other words, I said there's multiple subtypes of muscarinic and actually the alphas and betas, and what those receptors are linked to, right? And so remember I said here, it's ionotropic. In other words, we want to get graded potentials and action potentials. But out here, they tend to be, go back and look at the chemical synapse video, metabotropic. What we mean, again, by metabotropic is it's not necessarily opening ion channels, it's changing the metabolism, it's changing the function in a more prolonged and involved way than just changing ion fluxes across the membrane of the cell. That can be the result. Sometimes muscle contraction, and that may be what we need to do is cause ion fluxes and cause the muscle to contract. But even then, it's in a metabotropic way. In other words, it's not directly opening those channels; it's doing it indirectly. So anyway, we can change the activity, the metabolism of that target cell through differences in receptors and differences in what those receptors are linked to in the cell. Those are called second messenger systems or signal transduction systems. And again, we haven't really talked about those, but it's just a way to get a different response with the same neurotransmitter, right? Acetylcholine at multiple different locations can cause different things because of what the target cell, how the target cell responds to that neurotransmitter. Okay, so summarize for parasympathetic, right? It's our rest and repose or rest and digest. So it's going to be promoting digestion functions. It's going to be antagonistic to a lot of sympathetic responses at, like, for instance, the heart and other targets. The parasympathetic division stimulates growth and cellular repair, anabolism, building, anabolic things. That's a process of building. It is craniosacral in origin. There are long preganglionic, remember there's the ganglion, long preganglionic neurons and short postganglionic neurons. The ganglionic neurotransmitter receptors are the exact same as they were in the sympathetic division, but those at the effector or the target cell differ, right? Instead of it being norepinephrine, as it was in sympathetic, it's now acetylcholine. Instead of it being alphas and betas, which is what norepinephrine and epinephrine bind, it's going to be muscarinic cholinergic receptors, right? Muscarinic, the subtype of cholinergic receptors at the target tissue, right? And that's going to give us the parasympathetic effects at cardiac muscle, smooth muscle, glands, and adipose tissue.